Hello, everyone, and welcome to part one of the lecture on chapter 3, cellular structure and function. We skipped over chapter 2 on chemistry. We're just jumping straight to chapter 3. All right, a couple of questions for you, reviewing some material from chapter 1. What is the main structural feature that eukaryotic cells possess, but prokaryotic cells lack? You can pause your recording. And you'll welcome you back. Here are the options: cytoplasmic membrane, cytoplasm, nucleus, D, ribosomes. And if you thought to yourself the difference is C, the nucleus, you are correct. That is, so that's what the karyo refers to. Karyo is kernel or inner central core of something, and U means good or true. These are the cells that have a true nucleus, and pro means before. You may hear my cat walking around. Let's see. So C is the correct answer here. Which of the following is not a eukaryote? A, algae, B, protozoa, C, fungi, D, archaea. And if you're thinking to yourself, the one that's not eukaryotic, these are the archaea, that is correct. All the rest of these algae, protozoa, and fungi, they're all in the eukarya, domain eukarya. So let's get started. The overview of prokaryotic and eukaryotic cells. We're going to focus on prokaryotes first. Remember, there are two domains of prokaryotes. Those are the bacteria and the archaea. What they have in common is that they do not have nuclei. Although they do have genetic information, like eukaryotic cells, they don't store it away in a separate nucleus. In addition to not having a membrane around the nucleus, they also don't have membranes around smaller structures that eukaryotic cells do have. Things like lysosomes, for digesting or breaking down molecules. So there are some advantages to the eukaryotic cells. One is that separate nucleus, the other one is separate organelles, separated by phospholipid bilayer. They're minuscule. They're about 1 micrometer in diameter or smaller. That's why they went undetected for so long. It wasn't until the 1800s, when the microscopes got better, that people started to notice that there were these things that were so little. They're quite simple in structure. I think of these always as, like, if you know what a studio apartment is, it's just one big room. There's a kitchen on one wall, and there's, a place to sleep, but that might also be the living room. Little and simple. Here's a composite figure. The upper part of the figure is a transmission electron micrograph, TEM, and that's all in black and white. And then this part down below has been kind of colorized, cartooned, so you can see a little bit more detail. A couple of things. Here's the scale. So this length is half a micrometer. So it's about 1 micrometer across, and maybe 3 or 4 micrometers in length. Pretty small. This is hundreds of thousands times smaller than a eukaryotic cell. Some of the structures that we can see, let's see, where do I want to go? So cytoplasm, all of these cells have cytoplasm. That's the fluid inside the cell, and the materials are kind of suspended in this fluid. There is a cell membrane, a cytoplasmic membrane. That means the same thing. And outside of that, the prokaryotes have something called a cell wall. We will go into detail of what that structure is. Outside of even the cell wall, there's something referred to as a glycocalyx. Glyco sugar, it doesn't matter. Glycocalyx are on the outside. And there are ribosomes, just like in eukaryotic cells, there are ribosomes. It's an important principle. This, remember, is the organelle that allows us to conduct translation, taking information from RNA and using it to put amino acids together in the right order to make proteins. And then some bacteria have other structures that stick out from them. Now, eukaryotes, in contrast, that's kind of a defining characteristic of eukaryotes, is that they have a nucleus, a separated nucleus with with a phospholipid bilayer around it. And there are some internal membrane-bound organelles. So although prokaryotes and eukaryotes both have organelles like ribosomes don't have a membrane around them. The kinds of membrane-bound organelles I'm talking about are things like Golgi apparatus, lysosomes, separate compartments. They tend to be bigger, 10 to 100 micrometers in diameter. As far as volume goes, that makes it 1,000 times bigger than prokaryotic cells. Eukaryotic cells are enormous compared to prokaryotic cells. They're much more complex. As I said, the prokaryotic cells are like studio apartments. These are like, apartment complexes, lots and lots of little rooms, separate structures. And the kingdom, so it's the entire domain eukaryotes, one domain, but then there's multiple kingdoms within that domain. And last time I showed you that there's many, many, many kingdoms. For simplicity in this class, we're still going to lump all the many kingdoms into the majority of them. We're just going to lump them together into Protista. This will include algae and protozoa. They're single-celled organisms. Now, separately, we'll deal with the fungi, plants, and animals. Okay, here's a cartoon. Well, the upper part is, this one is a scanning, no, it is also transmission electron micrograph up here in gray. And then the rest of this cartoon is just colorized cartoon. A few things, here's the huge nucleus. This is where we keep the genetic information. But then there's a bunch of different organelles, like, for example, here are these sheets with little dots on them. The sheets are the endoplasmic reticulum. And the little polka dots, the red dots, these are the ribosomes that are helping us to translate RNA information to make protein into that endoplasmic reticulum, Golgi body, Golgi apparatus, whatever you want to call it. This is an important thing. The cytoplasmic membrane, this is that phospholipid bilayer. We're going to go into it in more detail. This we have in common with the prokaryotes. They also have that. That's all I needed to cover. 10 micrometers in diameter. I'm sorry, 10 micrometers is the measure down here. And you can see that's at least, say, 40 micrometers in diameter. So much bigger than prokaryotes. And this diagram, again, is to illustrate, to drive home this point. This is how small bacteria are. So we're going to start not with the smallest thing on here, but we're going to start with a particular bacterium. Here's one bacterium. It's 1 micrometer in diameter. Little, right? It took a long time to discover bacteria. It wasn't until the 1800s, like I said, microscopy hadn't gotten good enough to detect some of these items. And look how much smaller than that viruses are. That's why it took even longer to detect viruses and figure out what they were. We knew that the effects of viruses, we hadn't seen them yet. Okay. So you can see the bacterium looks enormous compared to the virus. Where is my, I keep losing my cursor, sorry about that. And then the bacterium looks really little in comparison to a eukaryotic cell. This is a protozoan that can cause disease in humans, Giardia. It's in the fresh water. It's the reason why people have to filter water when they're out hiking and trying to drink from streams. This thing's huge compared to a bacterium. And then again, to drive home the point of how little all of this stuff is, this square, if it represents this square here, this entire oval, that is a chicken egg. So what we are talking about is minuscule. And again, that's why not that we're dumb, we couldn't detect it. We didn't have the technology to detect things this small until the late 1800s and early 1900s. All right, some external structures of the bacterial cells. I mentioned the glycocalyx in passing. So glyco refers to sugar, and calyces, these are on the outside of the cell. It's gelatinous, sticky stuff on the outside of the cell, made of polysaccharides, those are sugars, polypeptides, those are strands of amino acids, not long enough to be full proteins, but made of the same stuff. And it's a way for, well, let's just talk about two glycocalyces. One is tidier, it's tighter, it's called a capsule. And you know what capsules are, like space capsules or medical like tablets. They're capsules that are filled with stuff inside them. That's what we mean by capsule. So they're very organized, repeating units of these organic chemicals, organic, again, carbon containing, like these sugars and amino acids. They're attached to the cell surface. And we think that these capsules may be in part what keeps bacteria from being recognized by an organism that the bacteria is trying to live inside. Then the other kind of glycocalyx has this very informal sounding name, but I promise you this is the actual name for it. It's called a slime layer. Why it's called that is because it's kind of slimy, and it's more loosely attached to the cell surface. It's water soluble, but it's sticky enough that allows the prokaryotes to attach to surfaces. The stuff that grows on teeth, remember, felt the kind of furry teeth in the morning. All right, some more external structures of bacterial cells. These have to do with locomotion. Some bacteria have flagella, singular is flagellum. These are responsible for how some bacteria can move around. So there are these long structures that extend well beyond the cell surface. Not all bacteria have them. What their function is to move. And so that it's kind of cool. They rotate right at like where they're attached to the bacterium. They actually can rotate there, and they can propel the bacteria through the environment. Sounds awesome. It's a little bit sloppy, to be honest. So bacteria, they can move in response to stimuli. The term that means moving in response to stimuli, that term is taxis. If you're thinking that looks like taxis, yeah, it does. It means the same thing. It's movement, moving from one place to another. It's not really, it's not perfect. Our textbook goes into some detail about runs and tumbles. I'm not going to go into all of that. Just one of the points that I want to make here is the bacteria can kind of tumble around and then sense what molecules are nearby, and it can detect, am I basically, am I moving? Am I moving in the direction of food or not? In other words, are the nutrient molecules coming in higher concentration or lower concentration? And so it can detect that, and then it can quick do this organized looking run where it can move in one direction, and then it stops and checks again. And so it's not a beeline. It doesn't make a straight line towards food, but it kind of can get itself in that, moving in that direction. Let's see, some cartoons over on the left. These are two different kinds of cell walls that we're going to go into later. What I wanted you to see is that the flagellum, the thing that's sticking out from the cell, that thing can rotate and where it rotates is here, where it's attached. We'll go into the difference between gram-positive cells and gram-negative cells later in this chapter. Let's see, a few terms. If it has one flagellum or a couple of flagella, or a handful of flagella, or a bunch of flagella, there are different terms for these things. So monotrichous, amphitrichous, amphi meaning having it the two ends, lophotrichous, having a tuft, like a ponytail of flagella at one end, and peritrichous or peritrichous, sometimes it's pronounced, having these flagella all over the place. And so here are some micrographs of these different arrangements. So a particular species of bacterium has one arrangement of these. Okay, some more external structures of bacterial cells. We have fimbriae and pili, or pili. So first we'll do with fimbriae. Fimbria is singular, fimbriae with an extra E at the end, that's plural. So the flagella, as you can see, they're longer, and then the fimbriae are a little shorter. They're sticky, multiple bristle-like projections, and they're used to kind of hold on to a surface or to each other. Their function is important in these so-called biofilms, like I was describing on the teeth, on the inside of your water bottle, if it ever goes for a while without washing. If it's kind of slimy, that's that stuff. Okay, some more external structure, pili or pili. This is pilus, singular. These are super cool. And the way to remember what they are, I think of them PI to remind me of pipe. This is kind of hollow on the inside, and this allows the cells to pass information from one cell to another cell. Genetic information can get passed along from one individual to another individual, and they're not passing it to the next generation. This is the same generation transferring genetic information to each other. I mean, that's extraordinary. So it's a special type of fimbria. It's also known as a conjugation pilus. Let's see. We'll cover this in another chapter when we talk about genetics. But conjugation is this thing that bacteria can do that seems weird to us as eukaryotes because we don't do it. But lots and lots of prokaryotes do it. And essentially they can get close, grab a hold of each other, and then pass little rings of DNA to their partner. And it's not like to make a baby. This is like, here's some genetic information that you can use in this generation right now. So if like you and your friend could pass genetic information that would help you navigate your environment. It's amazing. This is actually, we'll talk about this later, but this is how, like, for example, antibiotic resistance can spread in a population of bacteria within a hospital. It's amazing. Yeah, they typically have one or two per cell. It's not a huge number of these pilus. Yeah, so this is the function, is to transfer DNA from one cell to another during this process of conjugation. All right, a couple of questions to make sure that you're following along. Sometimes prokaryotic cells are surrounded by a gelatinous sticky substance. What is this structure called? Glycocalyx, cytoplasmic membrane, cell wall, filaments. And if you were thinking, that is the glycocalyx, that would be correct. Let's see, I can't actually read this question because something's blocking it on my screen. What is does it say external structure prokaryotic cells? Nope, I gotta move this. How am I gonna do that? There we go. What is an external structure of prokaryotic cells that helps them adhere to each other and to substances in their environment. Fimbriae, flagella, lipid A, peptidoglycan. And we haven't mentioned lipid A or peptidoglycan yet today, but we have talked about both fimbriae and flagella. And the things that are going to help them hold on are the fimbriae. And the flagella, remember what those are for? Moving through the environment. All right, some reminders. There were mastering assignments that were already due for chapter 1, or the kind of just check to make sure that you know how to use mastering, and then there was one for chapter 1 that was already due. There will be mastering assignment or assignments, plural, due next week for chapter 3. I haven't decided yet if it's going to be one assignment or two. And then we'll talk more about endospores next week. We're going to do our first exam in February. The exact date will be determined, and I'll announce that in advance. And it's going to cover material from the first day until the exam day. All right, I will stop here and pick up with part 2, where we'll talk about cell walls in prokaryotes. See you then.